Welcome to another episode from the Veterinary Research Channel. Today, we're diving into a fascinating study that explores new ways to detect cancer in our canine companions. Finding cancer early can make a huge difference in treatment outcomes, and this research looks at specific substances in the blood that might act as warning signs. The study, conducted by a team of researchers in South Korea, tackles a significant challenge in veterinary medicine: the need for an accurate and easily accessible method for diagnosing malignancies in local veterinary clinics. Currently, it can be difficult to get a definitive diagnosis without sending samples to specialized labs. This research aims to evaluate serum thymidine kinase 1 protein, or TK1, and its autoantibody as potential tumor biomarkers in dogs. TK1 is an enzyme involved in DNA synthesis and cell proliferation. When cells are growing and dividing rapidly, like in a tumor, TK1 levels tend to increase. The researchers also looked at autoantibodies, which are produced by the body's immune system to recognize and bind to overexpressed, mutated, or misfolded proteins released from damaged tissues. These autoantibodies have the advantage of being stable in serum and easily measurable. Let's get into the specifics of the study. The researchers collected serum samples from a large group of dogs, totaling over 1,700. These dogs were categorized into 4 groups: healthy controls, dogs with non-tumor diseases, dogs with benign tumors, and dogs with malignant tumors. The team then measured the levels of TK1 protein and TK1 autoantibodies in these serum samples using a method called lateral flow immunochromatographic assay. Think of it like a sophisticated home pregnancy test, but instead of detecting pregnancy hormones, it detects these specific biomarkers. So what do they find? The results showed that TK1 autoantibody levels were significantly higher in the group with malignant tumors compared to all other groups, including healthy dogs, dogs with non-tumor diseases, and dogs with benign tumors. Specifically, the median autoantibody level in dogs with malignant tumors was 0.71, while the median for the other 3 groups hovered around 0.34. This is a notable difference. Furthermore, the study broke down the malignant tumor group into different types: carcinomas, hematopoietic tumors, and sarcomas. Carcinomas are tumors that arise from epithelial cells. Hematopoietic tumors are cancers of the blood and immune system, and sarcomas are tumors of the connective tissues. The researchers found that TK1 autoantibody levels were significantly higher in all 3 of these categories compared to the healthy control group. The highest levels were found in dogs with carcinomas, followed by hematopoietic tumors, and then sarcomas. In contrast, while TK1 protein levels were elevated in dogs with malignant tumors compared to healthy controls in dogs with non-tumor diseases, there was no significant difference between dogs with benign tumors and those with malignant tumors. This suggests that TK1 autoantibodies might be a better marker for distinguishing between benign and malignant tumors. To assess the usefulness of TK1 protein and autoantibody, the team generated receiver operating characteristic curves, also known as ROC curves. This is a statistical method to evaluate how well a diagnostic test can distinguish between 2 groups, in this case, healthy dogs and dogs with malignant tumors. The area under the ROC curve, or AUC, ranges from 0.5 to 1, with higher values indicating better accuracy. The AUC for TK1 autoantibody ratio was 0.758, while the AUC for TK1 protein was 0.633. These results suggest that TK1 autoantibody is a more reliable biomarker than TK1 protein for differentiating between healthy and tumor-bearing dogs. So what does all of this mean? The study suggests that measuring TK1 autoantibody levels could be a valuable tool for screening dogs for cancer. The researchers acknowledge that neither TK1 protein nor its autoantibody are perfect as standalone biomarkers for detecting hidden or occult malignant tumors. However, TK1 autoantibody shows promise for differentiating between healthy and tumor-bearing dogs. The researchers compared their findings with previous studies that measured TK1 protein levels using different methods. They noted that other studies have shown TK1 protein levels to be significantly increased in dogs with lymphoma and leukemia. In solid tumors, the increase is less clear. However, with the development of more accurate TK1 protein assay methods, significant increases have also been found in gastric, ovarian, lung, prostate, and breast cancers. Interestingly, This study is among the first to investigate changes in TK1 protein autoantibody levels in dogs with malignant tumors. The researchers were able to demonstrate that TK1 autoantibody is useful in the screening and diagnosis of hematopoietic tumors and various types of carcinomas and sarcomas. The TK1 autoantibody ratio was also significantly high in lymphoma and transitional cell carcinoma, consistent with other studies. The team also looked at how TK1 protein and autoantibody levels varied in dogs with non-tumor diseases. They found that TK1 protein levels were higher in dogs with hepatobiliary disease, while TK1 autoantibody levels were lower in dogs with immune-mediated disease. The researchers hypothesized that immunosuppressant drugs, often used to treat immune-mediated diseases, might be responsible for lowering TK1 autoantibody concentrations. While this study provides valuable insights, the researchers emphasize that further investigation is needed. They suggest that future studies should include a larger number of dogs, subdivided by tumor stage, to re-evaluate the sensitivity and specificity of TK1 autoantibody as a biomarker. They also call for more research to elucidate the mechanism of the autoantibody and the interaction between TK1 autoantibody and protein. In conclusion, this study highlights the potential of TK1 autoantibody as a biomarker for detecting malignant tumors in dogs. While not perfect as a standalone test, it shows promise for differentiating between healthy and tumor-bearing dogs and could be a valuable addition to a multi-biomarker approach. By combining TK1 autoantibody with other biomarkers, we may be able to develop more accurate and reliable methods for early cancer detection in our beloved canine companions. Thank you for listening to the Veterinary Research Channel. A link for the referenced paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.